is the aim of all network. You see, this is have a nice biologist also. This is very interesting. You see, good biologist. That's very good, you know.
That sounds interesting. Sounds good.
Yes, that's the idea. Of course, for example, I want to invite you to be a member, né, this network, like consulting, shade the things, etc. Why one of my aim to invite you today is invite you to see what you do. Of course, you receive Rodrigo Profeta, and you have project with Aeromonas hydrophila, Brucella botus, sequence 52. You have Corynebacterium, tuberculosis also. You have one of my friends, Alexandre Rosado, we sequence 118 strains isolated from Antarctica, and he have a very nice collection, 500 strains isolated when he working in Antarctica, and that's a good thing, you know. That's a good thing, and you have the people with very good biology, the good question, the biology question. That's a very, very interesting to see.
Yeah, that sounds interesting. I mean, that's one of the things that's going now, I think, is, you know, lots of sequences are starting to become available. And when I'll say this in a few minutes, but when we started, sequencing was relatively difficult. Today, that's not so, but the biology is more difficult today.
Yes, yes. And now, Flavia, do you open for all people now, for accept all participants? Now you have 63 people, okay? And I will wait a little bit for more 2 minutes to start. And some people prefer use YouTube or the other media. Okay, no problem. And the question, you can, after Bart finish, you can do the question, and please write also for Bart in the chat, okay? And you open for the question at the end of Bart's presentation, okay? Yeah. You see Bart Weimer. I don't know what the mean C Weimer, but it's Bart Weimer. You see, it's Bart Weimer. He's you can see his profile. He's a professor in the population health and reproduction in UC Davis, Universidade da California, Davis. He did the PhD in 1990, a bachelor in Arizona, a postdoctorate in University of Melbourne, Australia. He's a PI in several projects, and you can find his information in the veterinary med ucdavis.edu. You'll see the name and all information about Bart Weimer. I'm very thankful Bart to give this speech today, this conference. Thanks so much, Bart. And of course, I will, in this moment, we organize the web page in Portuguese and in English. And I mean, I appreciate you really you accept this invitation to give a talk. He be member of all Omics Science Network. This network also try to find money in Brazil, okay, for all projects, all call. We will try to organize and do this. Try to find the money, for example. In 2 weeks, we will open a possibility to BRICS, Brazil, Russia, India, China, and South Africa. You try to organize a project together. You try also for the taxonomy call. We will open in 2 weeks, I think. You will organize to be together, and you at this moment with PhD student, master, PIs, you have 104 people, okay? And every week or every 2 weeks, we invite all the scientists. The next will be Jan Bombach from Germany. I will send to you the poster, etc. Okay, and the Flavia we include in all English information about all network. Thanks so much, and Bart, please start. I ask for all people close the microphone and wait at the end of presentation to do the question to Bart. Okay, thank you.
Okay, thanks, Bosco. Thanks very much for the invitation to talk today. It's a big challenge these days to organize a meeting, especially on Zoom, and everyone's becoming an expert at using Zoom. In Davis, we've stopped teaching in person from February. So now Zoom is our favorite thing to do. So let me share my screen so you can see the slides. Okay, can everyone see that okay? Because I can't, let me think here. There we go.
Yes, it's okay.
Great, okay. So, all right, what I want to do today is talk a little bit about the 100K Pathogen Genome Project, and it is the basis for a whole bunch of activities that have followed on since the 100K started. And I'll go through what the 100K is doing, but I'll tell you just a tiny bit about my lab before we start. So my lab is really interested at the intersection of population genomics and functional biology, which means a lot of what used to be called system biology. So I have two parts of the lab. We have an informatics part of the lab, and I maintain a wet laboratory portion. So we have a lot of different projects from, for example, we have a project right now with the USDA to look at microbiome of corn. And I'm not going to tell you about that project, but we have 2 papers coming out that are releasing about 1,500 bacterial genomes that have been isolated from a unique corn landrace in Mexico, which have novel nitrogen fixation characteristics. So we go all the way from plants and animals. We do a bit of food safety work, and we also do a lot of interactions and metabolism. So the context, is that not going to work? The context, so sorry, the context of what we're really sort of thinking about is how to use whole genome sequencing in public health, in some parts in food microbiology, but really it's outbreaks in public health. And to do that, having a large number of genomes became very obvious very early on. So we started the 100K project in 2012. And when I started this project, we were collaborating with the US government. And since then, the US government has, yes.
But it's better, I don't know if you can use the full screen, you see, and the PowerPoint full screen here. Yeah, yes.
So sorry.
Okay.
How's that? That works better. Okay, thank you. I totally forgot. So when we started in 2012, since then, the US government has implemented using whole genome sequencing for food safety, but also for outbreak detection. And that started in 2014 in the US. And since then, it sounds like a long time ago, but it's moved very quickly. And since then, there's been a large industrial consortium between IBM and Mars, the candy company, to learn how to use metagenomics and identify bacterial and viral isolates out of metagenomic samples. But what we realized very early on is that the databases the reference databases were inadequate. And so what we really needed was a large number of sequences. So in 2012, if you looked in GenBank, there were only about 2,500 to 3,000 genomes. And if you look today, there's over 500,000 whole genome sequences in GenBank. So sequencing has moved very quickly. The technologies have moved very quickly. The partnerships started and have come and gone. We formed partnerships in South Korea, China, Canada, and Mexico. And we're still looking for partners. When the 100K project has really focused now on using bacterial genomes really more for for disease outbreaks, and then understanding fundamental biology. So as I mentioned before, one of the reasons we got interested in this is because, as you all know, 16S sequences were used for really classifying bacteria and looking at relatedness. And so this tree that's on the left is showing you what a SNP tree is looking like for just a few isolates of Salmonella that are very closely related. And the dark red boxes, they should cluster together because they're the same species, they're variants of the same species. And yet they don't when you look at 16S. And at that point in time, when this tree was made in 2009, it was a little bit confusing because there were too few genomes. And so we started using what is called genome distance, or the total genome. Instead of a single gene, we started using the whole genome to understand how the phylogenetic tree would start looking. And so it looks much better. The variants cluster together much more closely by using the whole genome. And so that led us really into understanding that the more genomes we have, the better off we're going to be. And that led us to start the 100K project, which at the time was the largest project to be started. Today, there's other projects globally that are looking, doing other project tracking and other sequencing. But most of those projects are really focused on phylogenetic relationships and tracking genomes at a much smaller size number than we're contemplating. And so we're really looking for partnerships to sequence microbes from around the globe, sequence microbes from different environments, the same organism from various locations and environments and sources. So we've published a series of papers releasing genomes. And initially when we started, the mandate was to publicly release the genomes. To date, we've released about 25,000 genomes on GenBank. We have a much larger number of genomes that we cannot yet release. And so this listing is a few papers that we've published, and there's a few that are, like this paper right here is in review on Vibrio. That's going to release 150 Vibrio genomes, Vibrio parahemolyticus. And we've got quite a few other projects running with very large sets of data. As I said, the plant project will release in the first paper, release about 600 genomes. Secondarily, we've developed methods on how to use meta-RNA-seq or RNA sequencing to do metagenomics. And the purpose is not to do gene expression. The purpose is to identify the organisms and do relative abundance from very complicated mixed samples. So I've given you two examples that are in the public domain right now. One's published and one's on bioRxiv while we're waiting for the review to be finished. And these release about 50, 60 metagenomes, and give techniques about how to analyze them. So we've developed methods to take complicated metagenomes, and if they're from animals, for example, you sequence them and you can remove the animal sequences and then focus in on the microbial sequences. And of late, we've also been working on virus, as many other people in the world. We've got a paper on medRxiv, understanding genome variation and the transmission rate. So as we keep going, we're going to continue to release more and more metagenomes and more sequences. But I want to give you a bit of a perspective on what variation within a genome or genomes mean. So when we first started doing genomics, I've been sequencing genomes since 1991. And so back then, a long time ago, it was very difficult. It took about 3 years to sequence a single genome and about $3 million. So the first set of sequences that we ever did was a group of 12 genomes, and it took us 4 years to sequence them and publish them. And today we're sequencing, well, I just sent in a sequencing lane last week, got it back today, and we sequenced 3,500 genomes in one run of Illumina. So in a week, we have 3,500 genomes, and so they're coming out very quickly. So what does diversity look like? And if you look at genome distance, it's defined, a new species is around 17% different. So just in Campylobacter alone, which is found in lots of animals, it's found in environmental, it's found in food. In some cases, it's a commensal. It's can be a commensal in primates. 3% is very common in 700 genomes. In Helicobacter pylori, causes cancer, as you know, stomach cancer, 160 genomes, you have enough variation to have multiple species, certainly multiple subtypes. And if you look at Salmonella, the variation goes as high as 38% difference in the genome. And that's with 2,000 genomes that we analyzed. And so you can see those peaks show you that there's lots and lots of substrains beyond serotype variation within Salmonella. So all of this really led us to the perspective that we needed a lot more genomes. And the question then became, well, how many genomes do we really need? Because with a 3% difference and you're trying to identify a single organism, that only gives you 39% accuracy for a common identification. So if your genome that you've just sequenced is more than 3% different than what's in the reference database, you only have a 30%, 40% chance of getting the identification correct. So to achieve a 90%, 97% accuracy on identification, you have to have less than a 0.1% difference in the genome, which means, again, you need a very near neighbor or very close neighbor in the reference database to get the identification accurate. So the implication for that is, in clinical sequencing, if you don't have a near neighbor, you're going to get the wrong organism and the wrong diagnosis. So that becomes a very big challenge, which again says, for agricultural uses, it's important, but for clinical uses, it's crucial that you have very large and very diverse reference databases. So we asked the question, how many genomes should I be sequencing? Or when do I need to stop sequencing genomes? And so what we did is we created a calculation with our collaborators at IBM. And on the x-axis is the number of isolates. And on the y-axis is the number of new genes that we found with every single isolate that we sequenced. And so it's the number of new genes rises by a power law. So that tells you that you will never stop sequencing. So we did this with 792 Salmonella isolates from a variety of sources and said, okay, maybe that was an artifact of Salmonella. So we did it with collaborators with FDA and IBM, and we used 3,300 E. coli isolates, and the answer was the same. The more you sequence, the more new genes you find, the more variation you find, and it's virtually the same power law. And so we did it with Campylobacter with 17,000 isolates of Campylobacter. Same story. The more isolates you have, the more genes you find, and it's a slightly faster or a slightly larger power. So it means the evolutionary rate of Campylobacter is higher than Salmonella or E. coli. So 17,000 genomes of Campylobacter is a lot of genomes. And you would guess, or you would suspect, that that should be a big enough reference database. But in fact, it typically is not a large enough reference database in an organism that has a very high mutation rate. So I'll talk about this in more detail in a few more minutes. So the takeaway so far is that sequencing is never going to end, for good or for bad, because mutation rates never slow down, and you have unique isolates in different environments. So the need to sequence is going to continue, which then says we need to find ways to sequence very quickly, very accurately, and at a low cost, because we're going to continue to sequence for a very long time. But now it becomes important to know what to do with those sequences after we have them. They're not just to be putting into a reference database, which is what governments like to do, because they want to find outbreaks and they want to define outbreaks, and they need that reference database to be very large. And you actually saw this in action with the COVID sequences very early on in the pandemic. The small number of sequences that were coming in led to lots of inaccurate assessment of where the virus was coming from and where it was moving to. And now that the number of sequences are extremely large, you get more and better accuracy in identifying the source of the outbreak. So we wanted to ask this question in Helicobacter pylori, which, as you know, is important in progression of stomach cancer. So we did this all against all comparison, and this heat map shows you that there's distinct clades or distinct groupings within Helicobacter. Within Helicobacter. But even within a small grouping, there's a lot of genetic variation. And so we published a paper in 2017 that showed that you get infected with a polygenomic group of organisms, and as the disease progresses to cancer, you get a more unified or a more uniform genome, which demonstrated that your immune system selects the genotype that can lead into cancer. And not every genotype can lead to a cancerous situation. I'm going to show you some data, but Campylobacter does the same thing in livestock. So this, as I just showed you this, or said to you, this paper is showing that single colonies, you think a single colony is uniform within its genome, and in fact, it's not. It's very misleading to believe that you've got a homogeneous genome within a single colony. And this was shown nicely with biochemistry in the 1970s by John Thompson. And in today's world, when you start looking at single colonies, which we always strive for sequencing, you still end up with a large variation within each cell. So what was selected by the immune system that was related to invasion in cancer was really related to glycosyl hydrolases, the CAG island, which is already known to be involved in disease, and LPS, and also in additional neuraminidase binding proteins and enzymes. So a lot of genes related to the glycan is what was being highly enriched and selected for during disease, along with alleles of different virulence genes. We did the same sort of study in a broad sense in Salmonella enteritidis. There was an outbreak of Salmonella in sea mammals along the California coast, and this outbreak lasted about 15 years, and it was intermittent. And so all of this tree that I'm showing you, all of them have this identical pulse field gel electrophoresis pattern. So by classical methods that people use to identify these organisms, they look identical. In fact, when you start sequencing, they're not identical at all. And the yellow box shows you isolates that came from the California, the Pacific region within where we are. And they came from my group, in fact, with collaborators that found an entirely new clade of sea mammal isolates that were causing all the way from brain disease, brain inflammation, cancer-causing agents, and also gastrointestinal problems in a variety of sea mammals. But it was tracked down to originally coming from rats and horses that would defecate near the ocean and then get into the ocean, the isolates would. So with sequencing, again, the level of resolution at the gene level and at the SNP level can give you all sorts of new insights. There's no way we would have been able to do this without sequencing and without a large number of isolates. So new lineages are now being described in isolates that we would never have thought possible. So I want to give you a small perspective using Salmonella because it's relatively mutagenic, and the genome diversity tends to be high. So here's a relatively rare serotype called Schwarzengrum. And only in 15 in genomes, you can actually start identifying where the isolates were coming from. So if you start applying these metrics about percent difference, you can identify whether they came from the US or they came from Africa. And then you can go on and do other things that are much more interesting. But you can see these in a relatively small number of isolates, only 15. But as it grows, 59 Salmonella enteritidis, you can see where these metrics are now starting to place variation and changes in the phylogenetic tree, where you can identify where they came from, with whether it's a livestock source, a food source, or whether it's a human source, which is what's interesting for regulators and public health officials, because they can now identify source tracking. But in environmental samples, you can do the same thing. You can identify whether the samples came from the ocean, or they came from the forest, or they came from the soil. Salmonella Kentucky, one more, and then Salmonella Heidelberg, which tends to be in the US now, is starting to be associated with cattle and raw hamburger. So you can very quickly identify where isolates are coming from. So this whole point was, even with the small number of test isolates, if you have a very large reference database, you can get very fine resolution of information coming out. So then we went and asked the same question, just to be thorough. We asked the same question in Listeria monocytogenes, which is thought to be extremely low in diversity. But in fact, if you get enough genomes, 15,000 of them, which this is a couple of years old now, if you look at 15,000 genomes of Listeria monocytogenes, there's a lot more genome variation than you would have expected. There's at least 4 major groups, not subgroups, major groups. And there's many, many subgroups that you can identify within this plot. So now an organism that was thought to really be not very diverse, fairly monomorphic, is actually causing a lot more, but there's a lot more genome variation than you would have expected. Could you, if you had a sequence, could you get Listeria out of the identification? Yes. Could you get other interesting features? Probably not. And so because it's larger than we expected, we actually, I won't tell you about the paper, but we published a paper looking at the predictability of using whole genome sequencing and outbreaks and virulence. And we used packed biosequencing with 19 complete genomes. And based on just the genome characteristics, we were unable to identify specific genes that were predictive of virulence from outbreak strains. So there's still a long ways to go on what to do with the bioinformatics, but now we at least have enough genomes available to do some very interesting questions. But the caveat with that is, if you don't have the metadata to go with it, where it's from, what are the unique characteristics of the genome, you don't have enough handles to actually make a good comparison. And you're seeing this today with COVID. There's a next strain has a large number and a dynamic interface showing you the phylogenetic tree of every new isolate. So does the UK is also doing the same thing. They're sequencing thousands of isolates on microreact, and microreact gives you a very nice perspective of what the genomic distribution of COVID is across the UK and Europe broadly. And it's doing it also for the rest of the world. So if you are interested in what COVID sequences are like in Brazil, if you go on microreact, you can actually see what they are. But what no one's done yet is identify what's the variation going to have the impact on, especially in disease causing organisms. What is the impact for transmission or virulence? That's still yet to be done. So I want to walk you through a set of data and give you a perspective of what capabilities look like to just do identification, and then move on into what it looks like to do disease tracking and mechanistic activities, mechanistic assessment. So I'm going to go back to Campylobacter because it's a very simple system to look at. So in Campylobacter, we were looking at 3 species. And if you start from the left, panel A, panel B, panel C on the left, we used 90 genomes from 3 species. It was very easy to identify those 3 species using the sequence when you only had 90 genomes. When you added 220 or moved to 218 genomes, it became a bit more difficult. The cloud of genes that they shared diluted the identification and made it much more difficult to identify and segregate jejuni from coli from larvae. And the same is true when you move to 700 genomes. It became almost impossible to identify what species it was when you started adding multiple species and only had 700 genomes. So again, a bit of proof to say we need more for very diverse organisms like Campylobacter and Salmonella and others. We need a lot of genomes to be accurate. I want to show you the data for the 17,000 in another version of Campylobacter. So you can see a lot of variation and a lot of substructure within the serotyping of Campylobacter. So in the viral world, they have a concept called quasi-species. And in the bacterial or microbiology world, bacteriology world, we don't really embrace this idea of quasi-species. We thought that bacteria were much more concerted in what their genome looks like. But when you get enough genomes of a bacterial sequence, it starts raising questions of maybe bacteria also have quasi-species. And they have these intermediate variants that we don't really see all the time. So serotyping and other classification systems really don't have this level of resolution to identify what a quasi-species would look like, because we have artificially simplified the system that we're asking the question about. What that has done, though, is hindered us from understanding what gene variation is occurring for disease, what does antimicrobial resistance look like, and how does it change over time, and how many genes are really associated with these phenotypic characteristics that we need in the clinic and for identification. So again, how many genomes do we really need for functional links to disease? And that's what I'm going to venture into next and tell you about some work that we've done with large-scale sequencing, population genomic evaluation, and then asking questions about disease. So we published a couple of papers around Campylobacter and looking at zoonotic transmissibility. And we did it with the 700 genomes that I just showed you a minute ago. And we and a few other people in the world have had this idea that there is a generalist category that has a genotype that is permissive for zoonotic transmission. And then there's this host adapted notion, especially in Salmonella, Campylobacter, E. coli, where the genome somehow mutates and adapts just to that host. And that, again, you're seeing this play out with COVID right now, of host adaptation and the source of the virus that's perhaps jumped from a wildlife species to a human species. So what we found was interesting is as the genome evolved to be a generalist, AMR or antimicrobial resistance genes also segregated with variation and zoonotic transmission. Tetracycline resistance, without any selection for tetracycline, allowed it to move or segregated with genomes that were zoonotic. So we picked out a specific allelic variant or a specific allele and its population of a gene called CDT, which is a cytolethal distending toxin that's produced as part of the disease progression of this organism. So we picked this single gene and we started looking at the presence of this gene and where it came from and what animal. And we started identifying that there was no correlation of the genomes with the geography or the host. So what I just told you a few minutes ago about we can identify where the isolate is from is partially true, but if you don't have enough genomes, you can't make that assessment. So then we started focusing on just one type of gene, a pore. It's a porin gene that is also implicated in virulence in Campylobacter. So by using this marker gene, we could look at the identity or the allelic variation within single genes. So now I've taken you from whole genome analysis of what genes or what the allelic variation is across the entire genome down to 2 specific genes that are known to be virulence factors. And I'm showing you that there's actually allelic variation within these genes. And PORA is a core gene in the genome of Campylobacter, as is CDT. So within a core gene, you now have allelic variation, which actually explained completely the disease types that were occurring within the population of animals. And so you have some groups like these alleles that were found in crows and primates and in humans, they were not causing any sort of disease. But when you get down here, these alleles are causing disease in sheep and cows, abortion, in fact. But then this group also did not cause disease. So you've got at least 3 clades of alleles. So this actually in CDT, and this actually was very interesting and caught my eye very carefully. So CDT is well known to be a disease variant in Campylobacter, but PORA, there was a lot of debate of what's happening with PORA. In some cases it was present in disease, and in some cases it wasn't. So what we did is we took a very large number of Campylobacter genes and did a spatiotemporal assessment of allelic variation, and we applied machine learning to do that. So what we did is we took a classical GWAS assessment, but it provides a list of genes and alleles of those genes that are not ranked. And we actually started figuring out what alleles we tried to figure out what alleles were important. But the statistics that go with that are a little bit difficult, and the linear relationship and independence are really not true. That is what you would want in the statistics. So we applied machine learning after we did a GWAS assessment, and it provided a ranked list of features. We didn't have to know any prior assumptions, and it didn't violate any of the statistical relationships that we were interested in, which is great. But machine learning is data hungry, and you need a lot of genomes and a lot of variants, which this system created, because from one gene, we actually ended up with 1.25 million variants in the population that we looked at. So that was perfect for machine learning. So the way we set this up was the question we asked was, what gene or what allele of PORA was related to survival in intestinal tract, or became extra-intestinal and caused abortion? And so we set it up more or less a pairwise group comparison of isolates of PORA that were found in the GI tract and isolates of PORA that were found outside the GI tract. And after the scheme I just showed you, we ended up with, this is the top set of lists of ranked alleles, and all of them without us predisposing or skewing the data set to PORA, the top ranked alleles were all PORA, and they were all various alleles of PORA. And it gave us a ranked scale of importance. So we knew not only which alleles, but which alleles were most important relative to the disease. So luckily there was a published paper already that we could validate this approach to, and it came out that there were absolutely validated places within the PORA gene that were linked to abortion already. And in fact, we found those alleles and more. And then we did this across a 35-year span and asked, is this just a one-off type of arrangement, or over 35 years of doing evolution within the population, can we still find PORA alleles with genomic variation? And the answer is yes. You see this selected allelic variation. So it's standing the test of time, where if it's really going to cause disease, which is the bottom green lines, those are the alleles that are going to be the disease-causing alleles, and the ones in the upper part that are red are random variation within the GI tract. So this starts aligning up with the same story that we published for Helicobacter. Animals get infected with multiple genomic variation, And in fact, when it escapes the escapes the gut and the immune system starts selecting the alleles that are evading the immune system and able to cause disease. And we went on and further profiled the protein changes that occurred. And in fact, these changes, the top ranked alleles, resulted in 4 versions of the same protein with slight variation, which changed the activity of the protein. But those alleles could never be linked back to the entire genome. So this plot's a little bit difficult to understand, so I'll walk you through it for just a moment. The protein version on the left side is found in that's the poor A allele that's found in gastrointestinal tract, but it's spread across many, many genotypic variants of the Campylobacter genome. The three versions that are extraintestinal and cause abortion. So 3 separate alleles cause abortion. There's a common one that's often found, and it's found in this great big blue block, but it's spotted with this intermediate variant, which is actually almost a chimeric version of this protein, but it's spotted across all of the entire genomic variation. And then there's one that has a very large deletion, and it's only found in these small pockets down here. So the story so far is the all against all heat map shows you the genetic variation over the entire genome, but it's not predictive of specific allelic variants or specific disease-causing variants over a 35-year span. So while looking at the genome gives us some information, we have to drill in deeper and deeper and deeper to get to specific allelic variants to understand what's actually really causing the disease. So this is the track that we're on in many of the programs that we're working in, and we're actually working on this in COVID at the moment, and we're working on this in a variety of other organisms, including Salmonella, because it allows you to find new virulence factors and then scale the correct resolution to make sure that you get the mechanistic perspective that results in changes. But that's the good news. The bad news is, if you want to look at clinical setting, you don't know which scale yet to find, because if you look at just the entire genome, you get some amount of information like source tracking, are they related? But if you want to look at things like AMR or virulence, genome distance or even SNP maps are not going to give that to you. So we have a paper coming out actually that's comparing SNP maps and genome distance. And in fact, they give you slightly different information, but it looks so far that genome distance is going to be a bit more robust than SNP maps or SNP trees. So the point of this entire section is that from very large scale genomics, 700-ish genomes, you still have to get to a gene, and more pointedly, you have to look at the allelic version of that gene that's present in your population and understand if that's capable of causing disease in the situation that you're thinking of. So it's not just, is poor A present or absent, it's which version of poor A is present to cause the disease. Okay, so of course I can't leave without telling you a little bit about the work we're doing in COVID, because it's a merging of genomic variation and disease transmission. And this paper, what I'm going to tell you about, is available on medRxiv at the moment, and it's in review for publication. So what we did is we're trying to merge epidemiology with genomics. And if you look around, there's not hardly any papers that have really truly integrated genomics and epidemiology, at least in infectious disease. People are using PCR of a few genes here and there. They're looking for screens, which is fine. There's nothing wrong with that. So what we did is we set up this investigation to look at the index case over time and what happens to the genome over time and what happens to the epidemic outbreak curve over time. So if you go from the top left, you look at transmission rate, which is called R, the reproductive number, which is called R. And then you can derive that number out of the epidemic curve and the number of case incidences. Well, at the same time, you can start sequencing the viruses, which of course people have been doing for quite a while now. But what no one's done yet, and what we tried to do in this paper, is integrate a genetic variation index with the reproductive number, or the estimate of transmission of the virus, which then will give a lot of criteria both for public health and physical distancing, but it'll also give you perspective about what happens to the chances of obtaining a vaccine. So I have a small effort looking at how to use genomics to do vaccine strategy. But what I want to tell you about at the moment is how does it line up? How good is merging epidemic curve, reproductive number, which is transmission, and the genome? So this slide is showing you, this is the epidemic curve. This was actually from March and April. So very early days in the pandemic. So this is Thailand, Hong Kong, and the US. Epidemic curve by just sheer number of cases, and then this is the reproductive number by two measures, a 2-day serial interval or a 7-day serial interval. And if you let your eye merge back and forth between them, it's showing you that the 2-day serial interval or a 2-day transmission time is about accurate at that point, and it is replicating the outbreak curve. So when we started adding in genetic variation from COVID, we chose South Korea, Singapore, and Wuhan. And at that time, Singapore was controlling the outbreak extremely well. And this is the outbreak curve in the black line. And the blue boxes is showing you the genetic variation of the longitudinal score of the virus changes in the virus. In Wuhan, and this dark line is where, or the dotted line right here, is where they announced to the world basically that they've got a problem, and that they think the world is going to have a problem. So ironically, they had published sequences clear back in December, showing that the virus was circulating, and it was changing even in December of 2019. Sorry, this says 2020, but it really should be 2019. And then in January, the blue boxes or the blue lines are showing you that the virus is changing dramatically and the epidemic curve is going up. And then there was a change in the virus again, and the epidemic curve started to go back down. Same is true in Korea. Now, South Korea had a bit of a difference because it was essentially one hotspot at that point that really started the outbreak in South Korea and it was kept secret by a group that was a church group. So this is evidence to show you that the genome variation is going up and down, and there's some viruses that are extremely variant at 4, and there's some viruses that are hardly variant at all. But when you get to about 2, there's enough change in the virus that you're going to get a change in the epidemic curve. So there are papers out now that have actually showing that this is likely to occur as well. Other folks have shown that very much so that the genetic variation in the virus is changing the characteristics of the outbreak. But that was a very large debate. Early on, it was a huge debate within the virology community about what the variation means and which SNPs were occurring, but they weren't using population genomics to do that. And so this approach uses a population genomics perspective and is really helpful in identifying and understanding what's really happening to genetic variation. Now, going down to specific alleles, yes, it's possible. And in the spike protein, there's certainly alleles that have changed, that change the virulence of the virus. There's more to be found, and we're working on that at the moment. We hope to have a paper at least on medRxiv by the end of the month, if not before. So more to come, but there's certainly a linkage between transmissibility and genetic variation. But what's hindering this is the associated metadata. There's not enough metadata with the publicly available sequences to allow a lot of detailed analysis. So it's possible to do a very large scale population assessment of tens of thousands of viruses, but there's very little you're going to get out of it other than relatedness because the metadata is not rich enough. Okay, so I want to stop with one last vignette and example of how we're doing population genomics and this is starting to move into, this project is starting to move into machine learning with metagenomes. So a zoo came to us with a problem. They'd had chronic diarrhea in their primate population for 3 years, 3 orangutans and 3 gorillas. And they had isolated Campylobacter hyointestinalis, which is supposed to be an organism that is associated with pigs. And it's not been shown to be associated with diarrhea in primates. So we got samples. They sent us 8 genomes. We sequenced them. Then they started sending us other samples in a longitudinal screening. We sequenced the initial genomes and said, you know, they had already tried 8 rounds of different antibiotic treatment, none of which helped. And so the animals were still having intermittent diarrhea. We sequenced the genomes and said, you know, we're noticing that there's no tetracycline resistance genes. And for diarrhea, tetracycline is not indicated typically. So they were willing to try it. They did a 10-day course of antibiotics. All of the animals stopped having intermittent diarrhea. So then we isolated another set of organisms after the antibiotic treatment. And lo and behold, you know, we have all these isolates. They all came out to be Campylobacter hyointestinalis, but this table is here to show you that in some cases, it didn't come out to be hyointestinalis. It came out to be fetus. So if you, depending on which service you use or which reference database you use, you're going to get a difference identification. So it's very tricky. This example actually is saying to you, if you don't have a big enough database, you're going to get the wrong ID. So I was recently at a meeting where a friend of mine was using a Weimer-based approach called Kraken, and he didn't like it. And they stopped using it. And they were actually fairly disparaging of Kraken. And I quite like Kraken if you have the right database. And you can see that in every isolate, Kraken got a correct ID, and you can implement it locally, and you can build your own reference databases. MASH is a scalable version of Kraken, which also uses a hash matrix. And that hash matrix can be small or large, but a paper's been published using MASH showing that if you use the largest sketch size, the accuracy of MASH is extremely high. BLAST is great, but it's too slow, and you can't figure out which config to use. So while it's accurate, it's not going to be very helpful. Okay, so this is really to show you that algorithms and databases matter. So we also developed a way in this project to use a reference-free genome network to tell you about the diversity and tell you about the name of the organism. So this is matched up with a GWAS assessment and a reference-free assessment to tell you which clusters are clustering by who has them and what their characteristics are. So we started off using a pangenome, which was, to be very honest with you, a pangenome was not my favorite analysis before this project, because people were usually producing this graphic and then stopping, and it was unclear what a pangenome was really doing for you. But if you go beyond the pangenome and you go beyond some of the core genes, you can start identifying groups of genes that are either subspecies specific or prophage islands that are unique to specific sources. But then if you look again within the core genome, VAC-A is a gene that's not supposed to be in Campylobacter, but in fact, there's a very large truncated version in Campylobacter, and these isolates contained the transporter domain and a small region of the P55 domain. This was pre-antibiotic. This is post-antibiotic. So there was a large variation in this VAC-A, which is supposed to be a gene associated with disease in Helicobacter, a close relative of this organism. And yet, post-antibiotic, they all were the same. There was a couple of indels. But when you started looking animal by animal, it actually showed a lot of genetic variation. So there was duplications and deletions. The CDT alleles had rearranged and were truncated. And then you started having a total population change within the microbiome. So the names are not exactly important in this. This is 2 years. This is the diarrheal setting. When 1 month post antibiotic, they had similar sorts of, sorry, I drew a line. They had similar sorts of populations, but not completely. But 6 months post, they were completely back to normal, which is what you'd see in humans. That would be a representative of a more normal microbiome. In gorillas, the story's different. This is 2 years pre, or the 2 years related to diarrhea, 1 month post antibiotic, completely different, and then 6 months post, not quite back to normal, but pretty close back to normal. So now we've got this ability to look at genetic variation. We've got the ability to look at which alleles are moving around and causing disease, pulling them out of the microbiome, or having isolates. So in summary, what I want to sort of, just give some summary comments. What I hope to show you today is that whole genome sequencing is really going to be powerful in public health. But at the moment, you've got lots of sequences, you've got to figure out how to use them. And public health at the moment is really only using SNP trees to identify small changes and then putting in parameters to say within so many SNPs they're identical. But that's not exactly where, it's not the whole story. There's more to it than that. So the whole other thing I hope to show you is that public resources, if you use them alone, are not really quite good enough yet. You need to have a lot of genomes and a lot more genomes than we have today. So we and others have termed this genomic forensics. And so how can we institute a forensics platform on the genomic scale? And what I showed you, I think, was somewhat confusing to a certain degree, is which scale are you looking for? Do you want to look at the whole genome? Do you want to look at the gene? Do you want to look at the allele? So to implement a forensics perspective, we're going to have to identify which scale of resolution that we want to work with or need to work with. And then can you use metagenomics to estimate risk? And the short answer is yes. I didn't show you a ton of data, but we have more out there, and the short answer is yes. So I would say multi-omics and genomics is largely coming into its own very quickly, not just observational any longer. We can actually start making really good diagnoses and really good observations about what the mechanism is of disease. So I'll end there, and I want to thank you all for your time today and your patience. And I want to thank you, Vasco, for the invitation, and the organizers to get this together. It's a large effort in today's world. So I'll stop sharing, and I'm happy to answer any questions.
Okay, thanks so much for this wonderful speech, wonderful conference. But I have a lot of questions, but I open for other people now. And first, I would like to invite Aristotle to speak a little about our program. His bioinformatics program, the network is born inside of the bioinformatics program. And I would like first to Aristotle, please introduce a little bit the bioinformatics program to Bart and do your question.
Okay, thank you very much, Vasco. It's a great pleasure to have Bart. I met Bart in Campos do Jordão.
Yes, I remember.
last year, and we have a dinner with many people. So it's a great pleasure. Your lecture was fantastic. And one thing that when people began to work with pangenomics of bacteria, I am a microbiologist, but I work with fungi. And all the time I said to people, to prokaryotic or bacterial and archaeal people, pangenomics is what you must call a population genomics of bacteria, okay? Because it just scratch. you must you must study real population genomics, because all the population genetics and genomics came from eukaryotic organisms, and so sometimes we have a difficulty to transfer from one taxonomic group or to the other. It's the same regarding the viruses. So I'm very happy that you are studying this and to propose and make a real population genomics of bacteria. So that I don't have a question properly. I have just a commentary. This was my commentary that I want to talk. And I am just finishing my what I'm saying now. All these lectures that are part from the RECOM, that in English would be Omics Science Network, that Vasco has already explained to us, is hardware in the graduate program in bioinformatics of UFMG that I am now coordinating it. So it's a great pleasure to have you and to have this brilliant lecture regarding real population genomics, and try to to explore the most of the population genomics of bacteria. Thank you very much, Bart, and I am going to pass to open to questions.
Thank you. So while if anyone has questions, I want to make a comment about trying to do populations. So it sounds great to do populations, and it is great. But as a student, my students are always complaining because getting the files together and getting all of the sequences and all of the metadata together is actually one of the biggest challenges to do the population, because microbiologists are not used to thinking about organizing them in populations. We're used to thinking about 1 or 2 or 10 isolates, which is easy. But when you're trying to do 17,000 isolates with all of the metadata, you can't do that by eye, and Excel is a horrible way to do it. So you've got to figure out how you're going to actually do this. So it's an informatics problem again. And then when you get that done, that's the first step. So Aristotle's correct. We borrowed techniques from the eukaryotic world of pangenomes, which then we went on and did something else with. And then we borrowed GWAS, which we then went on and did something else with too. So GWAS is not a very common tool in microbiology yet, but I suspect it will be, as the way Aristotle mentioned it, because you need to be in this population perspective. So yeah, thank you.
Okay.
Question? Please, if you have some, you can type in the chat also and send the question. The people from need to move to this platform. If you see in the YouTube or the other, for do the question. But I have a lot of questions, and of course I want to invite you with the PI, for the people from the RECOM to do some brainstorm, but for the whole project, okay? You are now all consulting, one of the all consulting, okay? And yeah, and I use also the T-shirt from the Brazilian Society of Bioinformatics. And the question, Bart, is, what do you think about single cells? Because when you do extract the genome, you extract the DNA from colony, have 1 billion of cells, okay? Well, this is population genomics, you know, when you sequence, because you mix all DNA extract and you sequence that. What do you think is for the bacteria, it's better do single cells, or you can do work in the colony, is sequence this several strains. That's one question. I have more, but I will start with that.
So, you know, so single cell microbiology, when it first started, I couldn't quite understand why you would want to do that, because colonies have millions of cells and they have a mixed genomic genotype. So what is the meaning of having the sequence from just a single cell from one colony? So I still have that question, but what I'm seeing in single cell now is even in eukaryotic samples that they're sequencing hundreds of single cells and then they're mixing them back together. So they're trying to create populations within a cell mass or a colony, but they're doing it one cell at a time. Whereas you mentioned, if you sequence a colony, you have a mixture of genotypes. The problem is we can't tell. What we can get out of that is the frequency of, it's either a SNP or the frequency of occurrence of any single base in that colony. But what we don't know what to do, or I don't know what to do with that, is how to treat that into disease. And so my first attempt of doing that, something with that, was what I showed you today in the Campylobacter abortion story of allelic variation in a core gene is still having a profound effect on the disease type. So certainly gene presence absence is not good enough. And so when you start getting at single cells, then I really struggle with how to take that information and apply it to disease.
Okay, thank you. Thank you. I have some question. Alexandre, okay, just a second.
I have a question, Mateus.
Yeah. Yes. Okay, Mateus, please do that. And I think, yes, please, Mateus, do the question.
Okay. Thank you, Dr. Weimer, for your lecture for us. It's very illustrative and very interesting for us. How do you think about the use of the study of the population of microorganisms from the same environment, using many bacteria from the same environment and evaluating with the mutation rates that you have studied some effect about the virulence of the whole organisms or specific pathogens. Did you see some study before or not?
So from the same environment, I still think, so what we've seen in this plant project we're working on, we have isolates from exactly the same sample, yet the genotypes are different and there's allelic variation, and there's still variation in the physiological capabilities. So I don't think that's counter to what you would expect if you did classical physiology. You get multiple isolates, they have multiple characteristics. Now what we're really seeing is the genetic variation within those that have the same characteristics are higher than we expected. So even from the same environment, you're going to see a lot of strain level variation well, at least in the projects I've seen, and I've seen some papers in the gut microbiome of humans. There was a paper that came out, I think it was from Pierre Bork's lab about 2 weeks ago, really asking the question about strain level variation, and host changes on the host from the microbiome was all related to strain variation within the microbiome. So on the chat box, that's more or less the same question that
yes,
was on the chat box too.
I think Bart, you know him, is Professor Rosado. Now he's in California also. You met him, no? Alexandre Rosado?
I think so. Yeah.
Alexandre, if you want to do the question, please open your microphone.
we're in different departments, right? So his question also asks about soil, aquatic, and extreme environments. In extreme environments, we sequenced a bunch of metagenomes from the Great Salt Lake in Utah, which is like the Dead Sea. Has a varying salt concentration, and in fact, we saw a lot of strain variation. And the strain variation was really from the isolate variation. So, yeah, I think, you know, both what Mateus and Alexandre asked is, you're still going to see the strain variation, and in my estimation, you're going to see a lot of genomic variation as well.
Okay, I have one other question in the chat, but is Raquel do the question? Yes.
So
How?
yeah, go ahead.
No, please answer.
Raquel asked, how would it help to sequence more genomes and learn new resistance mechanisms with the search for new antibiotics and ways to fight bacteria? Starting from knowing the genome, how can we carry this knowledge and improve diagnostic and resistance in the hospital? So that's a great question. That's a great question. Antibiotic resistance, we are finding new mechanisms. So I didn't show you the data today because we don't have it completely assembled where it's useful to view. We just finished sequencing a 35-year retrospective study of Salmonella with specifically to ask the question, are the antibiotic resistant genes that are important today, were they present 35 years ago in animals? So we got 600 isolates from mostly cattle, and then asked the question, do we find the same occurrence of antibiotic resistance genes in the 1980s as we see in the last isolate we had was from 2019? And the answer is antibiotic resistance genes were stable from 86 to 2019. The occurrence was stable, but the MICs are going up. So the question you're asking, I think, is if the genes are stable that we know about, how is it that resistance MICs can go up? What are there new mechanisms? Are there other alleles that are involved in the resistance that are changing the MIC? So I think my opinion is doing longitudinal studies of genomics is a robust way to understand not just evolution of the genome, but evolution of the traits and new genes that are starting to participate in activities that we weren't normally thinking about, like antibiotic resistance. So that's the way we're tackling it, is to do longitudinal studies and then matching it up with with antibiotics, matching it up with MICs.
Okay, Mateus, I have a question.
Okay.
Mateus? Mateus?
Sorry. Are you listening?
Okay, I open for Mateus, Adriano Barbosa after. Okay, thanks Adriano to watch Bart. I appreciate. Mateus, please.
Okay, thank you, Dr. Weimer, for the first answer. And I have another question related to antimicrobial resistance too. Do you think that we can use some machine learning tools to study a few genes, because we see many phenotypic variation in the antimicrobial susceptibility tests, mailing with serial dilutions tests, and sometimes we stood some point mutations that are very involved in the breakpoints changing in the bacteria. You think that there are interesting tools to see these point mutations in the specific genes in relation with the whole genome of bacteria, because we see the phenomenon of persistence and others that are associated to a high mutation rate in the whole genome, producing new genes of resistance and something. Do you think is interesting or not?
I do. I think it's exactly the right question to ask. So as I showed in the Poiré data, that the genome variation is not directly linked to the mechanistic, the gene of mechanism, the mechanistic gene and its variation. So there seems to be a lot of independent mutation rates occurring within the genome. So you've got this summation over the genome distance. Using genome distance, you have a summation of the changes. But when you look at each of the individual genes, which we already know are mutating and evolving independently most of the time, even within an operon, you can get independent variation of the genes within the operon. So for antibiotic resistance, I do think that you're going to be able to use machine learning and find new variants that are enriched within the population of genome possibilities and that lead to higher MICs. So we're trying to do that right now with these 600 salmonella that we have. But one of the problems is if you don't have enough data, machine learning doesn't work very well. You really need millions of data points to get machine learning to work. So we've also applied random forests to some other, it's machine learning, but it's a classification style machine learning. And so random forests will give you some amount of information, but you need even to get beyond random forests to look at antibiotic resistance and get to the specific allele. So in the Poiré story I told you about, we didn't use random forests. We tried it initially, but it gave us so many hits that we had to find a way to segregate them when we used a different approach for machine learning to get the ranked, we used a different approach to get the ranked variation. And I think it will work very well for what you talked about. I think antimicrobial resistance will be a really lucrative section of data to apply machine learning. Yep.
Okay.
But you need MIC to go with it.
Okay. Thank you.
Yep. Adriano has his hand up on the screen, so maybe you want to ask your question.
Thank you very much. Thank you very much, Bart. Yeah, don't worry. I'm a basketball player. I used to. So I used to be part of the bioinformatics program, and actually, I've never stopped collaborating with people in Belo Horizonte. It's really good to be joining this group and watching your talk. It was very, very interesting. My question is regarding hi Vasco and hi Flavia. Hi. So I know them. Hi. So my question is regarding the co-evolution of microbial communities. It was something that I would find very interesting to see on your slides. I saw that you have a lot of comparison across genomes of the same species, but then when you think about, for example, in Brazil, we have problems with environmental pollution, for example. And then if different communities, different species of bacteria, if the genes, they change in a coordinated manner, I would like to know whether would this be possible, this kind of signal to be possible to be observed by using your genomic comparisons techniques. That's my first question.
Right. So I think you're right on to a really good topic. It's a great question. So coevolution of genes within a community. So coevolution of genes within a microbiome, for example. So one of the papers that we have on bioRxiv at the moment is in review. We stopped short of doing the coevolution question. And so the data are sitting there. They're soon to be in the public domain. So yes, the answer is yes, you could certainly do a coevolution. And if you did it in a longitudinal study, it would be even more powerful. So you've hit the nail, so to speak, you've hit the nail on the head. In the gorilla study, we have a bunch of microbiomes in a longitudinal assessment. So we'd like to look for coevolution of genes that went with disease and no disease. So it's not just one single gene, but what are the group of genes? So I think the question that you asked is incredibly important. but the dimensionality of what you have to look for is so high that you have to find the right statistical framework to use.
Yeah.
Because one of the problems is machine learning. Machine learning would be beautiful to use, but machine learning over time, a time series machine learning algorithm doesn't exist to my knowledge. And that's what you'd really need to do a good job of what you talked about.
Yeah, great. Thank you. Well, if I would I have a second quick question. I saw your very interesting work on COVID. It's start, and I hope the paper gets out soon. Would be to associate the microbiome or the metagenomics or even the metatranscriptomics of patients to the severity of disease.
Yes.
Do you have any thoughts on that?
Yeah, I think it would be a fantastic study to do. So there's only very few there's very few transcript studies out. And unless you set up the transcripts properly of the beginning, the end, or the beginning, the middle, and the end of the disease and recovery, I'm not sure you're going to be able to make a ton of sense out of that. But you're asking exactly the right question. I think you will be able to understand what are the genes evolving in COVID itself, and what are the genes responses in the host. So one of the reasons I like to use RNA-seq for metagenomics is you can look at the microbial expression and you can look at the host expression simultaneously in the same sample, and you can make really robust conclusions of what the host is doing in response to the microbe. We've got a paper out where we did exactly that with stem cells, infecting stem cells with enteric bacteria. And it was really robust to see what the stem cells were doing, what the microbes were doing simultaneously. That was in vitro. What you need is what you're talking about is an in vivo study during an infection. So I think the techniques are now coming to the point, the sequencing techniques are coming to the point we can actually do that. But the analysis tools now are, falling behind just a little bit, because we need more robust algorithms to do these large data sets. But yeah, you're spot on. Those are great ideas and great questions.
Thank you very much. It's a very impressive talk. Thank you very much. And
Thank you.
bye to everyone.
Adriano, please, please say, Adriano is former PhD student in our program, and please, Adriano, where are you now? What you do? 1 minute, please explain what you do now for us.
Oh, sure. Well, I used to be a PhD student of the bioinformatics program. And then after my PhD, I went for some
Uh-oh, you cut out.
Adriano have some problem. Adriano? Yeah.
Hello.
Okay.
Back.
Yeah,
No.
Adriano, please do it. Explain it again, please.
Yeah,
please.
very quickly.
Yeah, I received the support of the Brazilian Ministry of Education in the time I was in Brazil to perform a PhD, like an internship at EMBO in Heidelberg. And then afterwards, I became an associate professor at the University of Paraná, working with bioinformatics there. During that time, it was very interesting because we could make a kind of a joint bioinformatics program between the Paraná Federal University and the Minas Gerais Federal University, which was a unique opportunity to, get students, supervised by professors in different institutions. Well, that was quite nice. And then I moved to Luxembourg. In Luxembourg, we worked in the scope of the translational biomedicine programs of the IMI, which is the Innovative Medicines Initiative here in the European Union. And it's a work co-funded by the European Pharmaceutical Industry Association. So, and now I'm working at the Queen Mary University of London. I'm pretty much involved with the NHS response to the COVID-19 pandemic, and also working with the UK Biobank folks together with Health Data Research UK. So we have a great interest on the topics that Bart presented there. So at Bart's, which is the name of the school that I'm part of, it's called Bart's in the London School of Medicine and Dentistry.
Yeah.
So we are very much interested to learn how the microbial variability on severe and like light non-severe case of COVID-19 they diverge, and how we could take advantage of a modulated microbiome in order to improve the response or reduce the risk of disease in some subjects. So that's a brief overview of my work.
Fantastic. So I was working with some folks in England when COVID hit, and they were, you know this too well, that the government came and said everyone's going to sequence COVID now, and it's done a great job. The MicroReact data repository is fantastic, and so I think England's actually got a very nice response to sequences of COVID. And the question I don't know is, how much metadata do they have that go with the sequences so that you could do the analysis?
It is actually, it's a great question, but because now there is a new initiative by the NHS. Actually, it's by the National Institute for Health Research, NIHR, and they have an initiative called HIC-COVID. And HIC-COVID intends to collect all the primary and secondary care data for the patients. Yep. And in case these patients that develop the COVID-19, we will have an opportunity to look on a retrospective manner on their health status before the diagnosis of disease. And eventually we can come up with some machine learning to predict the risk of severity, for example.
Excellent. That's fantastic.
Yeah, actually, it's a very interesting initiative. And if you have data to share with them, they allow you to access the full resources of data sets collected in different countries of the United Kingdom. So it's quite nice.
Right.
And just to make a bit of marketing as well, we have a memorandum of understanding with Federal University of Minas Gerais, and I'm sure that Vasco is aware of that. And this allows students exchange between UFMG and the Queen Mary University of London. So if there's people interested, we should talk offline about this.
Now, I can't do more anything. It's only Aristotle, because he's the boss, you see?
Okay.
No joke, but Aristotle is the bioinformatics program, the head of bioinformatics program. But it's one question, not an invitation. Okay, you can help you also in the all network. You see, you can be a guest, you can work together, and Ana Ibenco is the point is with us also. And in few days, we will open our web page will be ready. You can see all information also, Adriano.
Sure.
But I have two questions.
Thank you.
And one question, Bart, is, for example, what do you think now, for example, whole genome sequencing to metagenome for several diseases you see? The question is, it's very, and compared with 16S, but the question is, what do you think? Is the pipeline is ready to do faster or not? That's one question. And what do you think about for the answer to the question, if you avoid 16S now or try to develop more pipeline to analyze whole genome sequencing and metagenome, whole genome sequencing? In the last question is, when a few years ago, I asked for the guy, for the head of Gold Genome Online database. I don't like really draft genome. You open now is draft. And the guy said to me, it's better to discover gene. But after 2 studies of genomic population, study, for example, the evolution, drug discovery. You don't think you go faster but bad? Because the information, the bad information, the wrong information is jump for the other genome, jump for the other. What do you think about these two kind of things I ask you?
Yeah. So I'll show my bias a bit. I'm very underimpressed with 16S sequencing. I never do it. There's too many dependencies and there's too much variation. If you don't get the right primer set, you miss the organism. So I don't ever do 16S. So we don't do anything with that. The second question you have is related to genome quality and draft genomes versus maybe closed genomes. So I'm actually just working on a project right now where a collaborator sent me a genome that assembled into 1,600 contigs and wanted me to do a comparison with a genome that assembled into 27 contigs. And this is a small 3.5, 4 meg genome. And the answer became, I did it, but I didn't want to. And when I showed him the results, it was the difference between, they were the same genome and they were sequenced at 2 separate levels of quality and they looked completely different. So yeah, I think draft sequences are perfectly fine if you set quality metrics of a certain standard. And if you're below that standard, you throw the genome away or you sequence it again. But if you're above a certain standard, then draft genomes are going to be pretty darn good because of the cost, right? We sequence with Illumina, we're sequencing draft genomes for a very small amount of money now. Like the QC of the DNA in the library costs more than the sequencing process itself. So the point I'm coming to is if you have very strong quality metrics on the genomes you're going to use, then I don't have any problem using a draft. And in fact, Adriano made a point about COVID and sequences. In fact, none of the sequences that are available in strain have quality metrics. So the quality variation in the COVID sequences that are coming out right now are extremely high. So and that's actually leading to a lot of misinterpretation of the data. So what's really interesting at the moment is that because of COVID and because they're using genomics and they're relating it to outbreaks, the general public are now really starting to understand if you have crummy sequence going in, you're going to have skewed perspectives coming out and you're going to actually get things wrong. And we've watched countries get things wrong and then they have to go back and say, whoops, just kidding. It's really like this. So I think using sequences for politics is a bad idea. Unfortunately, my country's tending to do that at the moment. But if you get the scientist in there and say, have a set metric, then you're fine. And I'll give you a second example. It was a little bit long-winded answer, I'm sorry. The second example is when we were collaborating with IBM in 2017, we downloaded all of the entire of the SRA. And then we realized about 35 to 40% of the SRA, the quality of the sequence was so bad you couldn't use it. So at that time we had 320 genomes and we ended up only using 150 of them because the sequence quality was so bad, we threw the rest away. And so if you put strategic parameters in place, then draft genomes I think are okay. Until having a closed genome becomes cheap enough, draft is probably going to be okay for a lot of good conclusions to come out of that. Okay.
Okay. I have another question in the chat, please.
Yep, Jose. Jose asked a question. Jose asked, thinking about core genes and core genome, the quasi-unvariable part of the genome, how much variation could be expected within a species or between sibling species? Are there any cutoffs? There are cutoffs. In fact, there's a German group who have created cutoffs of what it is to be a strain, a species, and a genus. using not the core genome, but genome distance. And so DSMZ hosts that pipeline from the German scientists. So if you go on DSMZ and look for genome distance, you'll find the cutoffs. I don't remember what they are in the top of my head for what they use, but 3% difference is enough to be a new species. So they can give you all the details, but so 3% genomic difference. The other piece I didn't show you, which we've been looking at carefully recently, is all these metrics I just talked about: genome distance, allelic variation. If you go and do, you're familiar with MAV. MAV is a condensed version of arrangement across the chromosome. Genomes that have very, very high identity by the metrics we were just talking about, if you look at them with MAV, they've rearranged. They haven't, the genome's rearranged, but it hasn't mutated, so to speak. You don't get SNPs, but you get blocks of genes moving, and it's noticeable. So the question I have in my own mind, and maybe someone can help me, is if the genome looks identical with SNPs and genome variation or genotype, but it's got a rearranged genome, is it the same genome? Then what happens? Because MAV is showing you it's rearranged differently and it's in half the genome's inverted. So Vasco's done a lot of work with Lactococcus, and Lactococcus does this extensively. Lots of gram-positives do this extensively. Large sections of the genome just invert, but they look to be the same organism. So is it really the same organism if the genome's inverted? Don't know.
Yeah. Then I think I maybe have a time for the last question. But Bart, okay. Well, I think that I have space for one question, please. And the Bart, all team in the RICOM, you have one team sequence the Brazilian COVID genome. Now they produce a nice paper. I sent to you in your mailbox. It's about epidemiology, about sequence COVID in Minas Gerais state. Okay. The next time, today, the Professor Alcantara can't join us, but the next meeting, I invite you to brainstorm together with the PI of the project PI. I invite him, okay? And thanks so much, Bart, for this wonderful Weimer conference. And I appreciate a lot, and I learn a lot. And I need to see again. But all people, they will record this presentation. Everything is saved now. And Flavia, please show the next speaker, and maybe also you can send it to Bart all conference you will do in the next month. Mariana, please explain a little bit who is Jan Bombach, what they will talk in the next conference.
Hey, everybody, can you hear me? No?
Yeah, not see
yes?
you. Yeah.
Okay,
No.
So can you see me now?
Yes, now it's okay.
Okay, so it's my pleasure to let you know that Professor Baumbach is going to give us a talk in the 21st of this month, if I'm not mistaken. And I worked with Professor Baumbach in Freising for 1 year and 9 months and in Germany. And he's going to give his talk telling what he learned from Arnold Schwarzenegger, which is very interesting. So I think all of us are going to have a great time. And they recently developed a very nice work on COVID, and for sure he's going to talk about this work as well. And yeah, it's that.
Yeah. Okay, all people will be welcome to the next next conference. And thanks so much, Mariana, Flávia, you all organized the bioinformatics program. And Bart, thanks a lot. And now I will close the room, the conference room. Thanks so much, Bart, and I will be in touch with you soon, okay?
Great. Thanks to everyone. It was really nice. Appreciate
Thank you very
the
much. Bye.
opportunity.
Thank you. Thank you. Take care, my friends.
See you later.
Bye-bye.
Thank you. Bye-bye.
Bye.
Bye.
Thank you.
Okay.